DNA, structure, replication, recombination, and enzymatic metabolism. So this lecture is really going to be an introduction to DNA. It's going to be an overview of the different aspects of DNA, DNA structure, DNA replication, the components, and so forth. I am going to provide supplementary videos because a lot of the concepts related to DNA are going to be review concepts, but in those supplementary videos, you will get kind of the review from the lecture that I would typically give to my general microbiology students. So I'm just going to post those videos along with this one, and you can kind of go through those if you need that review. But deoxyribonucleic acid, DNA, is a genetic information storage system. It's a polymer of nucleotides. Nucleotides are phosphorylated nucleosides. Nucleosides are composed of a ribose sugar and a nitrogen base. There are 4 common nitrogen bases, 2 with a purine ring structure, and that's adenine and guanine, and 2 with pyrimidine ring structures, thymine and cytosine. The order or sequence of nucleotides in the DNA polymer is the code for information storage. So this is what a typical nucleotide looks like. So you have the sugar, the phosphate, and the base. These are the 4 bases: guanine, adenine, cytosine, and thymine. And you can see the base structure, these are smaller in structure. Those are the pyrimidines, and these are larger in structure, the purines. Now you do need to know which ones are purines and which ones are pyrimidines. The way that you can kind of distinguish these easier is cytosine and thymine both have the letter Y in them, and so does pyrimidine. So that's how I kind of keep that straight in my mind. DNA exists as 2 polymers joined together by hydrogen bonds. So hydrogen bonds hold the adjacent strands of DNA together. Between thymine and adenine, there are 2 hydrogen bonds, but between cytosine and guanine, there's 3. And these 2 polymers twist to form a double helix, sometimes known as like a spiral staircase type structure or an alpha helix. Polarity. So DNA strands have polarity based on the phosphodiester bond. DNA strands have a 5' phosphate end and a 3' hydroxyl end. So here is the 5' end, and here is the 3' end. Now, when a new base were to be added here, it would come in this direction, and the 5' end would be added in here, and the 3' end would be down on that end. And this shows what I just kind of talked about. So a new DNA strand is polymerized from the 5' end to the 3' end, reading the parent strand in the 3' to 5' direction. The replisome is a group of proteins assembled for DNA replication. This is a diagram of the replisome with many of the key players listed here. So we have 2 strands, which are called parent strands. Those are going to be separated. And when they're separated by the enzyme helicase. And then you have single-stranded binding proteins bind to each separated strand of DNA so that those strands don't come back together. Then you have DNA polymerase that starts to bind and make a copy of the genetic material. Now on the lagging strand, or the leading strand, it is synthesized in the same direction that helicase is opening up the DNA. So it can be synthesized continuously. But on the lagging strand, it waits for a segment to be opened up, and because it has to be replicated in the 5' to 3' direction, it actually has to be built backwards. So it's going to be produced backwards and in chunks. Those chunks are known as Okazaki fragments. So in order to start an Okazaki fragment, first RNA primase is going to put down an RNA primer. Then from the RNA primer, DNA polymerase can bind, and that's DNA polymerase 3 that would bind, and it's going to then synthesize DNA backwards to the direction of helicase. Then when the next section opens up, Another RNA primer is going to be laid down by primase, and DNA is going to be synthesized backwards. At the end of all this, you're going to have one continuous strand for the leading strand, and the lagging strand is going to have a bunch of Okazaki fragment chunks. Those chunks are going to be processed in a few different ways. So DNA polymerase 1 is going to get rid of the RNA and replace it with DNA. Then all of those Okazaki fragments are going to be glued together using DNA ligase. So this is what that looks like. You have the Okazaki fragments, and eventually the DNA polymerase 1 is going to replace those RNA primers RNA primers with DNA, and then all of these segments are going to be glued together. The two strands of resting double helix are known as being anti-parallel, meaning that they are produced in opposite directions. So recombinant DNA technology is talking about cutting apart DNA and putting chunks of DNA in different organisms. So it's taking DNA and moving the DNA around to different organisms or different cells. 3 factors were required to perform recombinant DNA technology. Nucleases cut DNA into pieces. Ligases paste the DNA fragments together. And plasmids carry the recombined DNA into the cell. There's some DNA metabolizing enzymes. Polymerases catalyze the formation of phospodiester bonds. Helicases unwind and untangle. Primase synthesizes a short RNA primer to prime the DNA synthesis. Methylases add methyl groups to nitrogen bases. And deaminases take amino groups from nitrogen bases. Nucleases can cut DNA. Ligases catalyze the formation of a single phospodiester bond. In other words, they can glue pieces of DNA together on the backbone. Exonucleases remove bases from the ends of DNA strands, while endonucleases cut DNA strands internally. There's a series of restriction endonucleases that we have discovered. And they've been isolated from E. coli, and you can see one from Haemophilus influenzae, and there's all different kinds that exist. But restriction endonucleases can be thought of as like mini DNA scissors. So they will cut in very specific sequences. So they'll go along a strand of DNA, and when they read a very specific sequence, they're going to make a cut at that point in the sequence. And that's what they're programmed to do. Where did we discover these from? Bacteria. Bacteria, just like every other cell on Earth, is prone to getting viral infections. So yeah, bacteria can get sick with viruses. Those viruses are called bacteriophages. The bacteriophages don't make people sick, they make bacteria sick. Their goal is to get inside of bacterial cells to make more bacteriophage particles. So, if this virus wants to infect a bacterial cell, step 1 would be to inject in its genetic material into the bacterial cell. Then it shuts down all the bacterial cell's normal functioning and turns that bacterial cell into a viral producing factory. Very similar to how any virus would infect any other cell. It shuts down normal cell function and forces that cell to make more viruses. Well, the incoming DNA is a problem, or viral genetic material. Could be DNA, RNA, single-stranded, double-stranded. Lots of different combinations exist depending on the virus that we're talking about. But when the genetic material is brought into the bacterial cell has evolved to be able to attack that incoming DNA. So when foreign DNA comes in, these endonucleases are present, and they can chop up the incoming DNA. Well, we figured out that bacteria evolved the ability to have these, and now we use them for all sorts of different recombination technologies. So they cut double-stranded DNA in different ways. So it can cut it in an overhang, or it can create a blunt end. DNA ligase would then glue complementary pieces or strands of DNA together. And plasmids. So plasmids are specially designed DNA transporters. So if we want to put a gene into a particular organism, we can cut the gene out of the host organism, we can insert it into a plasmid, and then use the plasmid to shuttle it inside of a new cell. This whole process is something that I did for many, many years. I made recombinant DNA in a research laboratory at UW-Madison. So I will explain this process in a little bit more detail in those supplementary videos that I provide to you. But this is basically it. Just a brief overview of DNA and the enzymes that impact DNA. We'll get into a little bit more detail in those supplementary videos.